All right, so we see a pretty cool cell right here. It is dividing. You see these individual cells, but here we got one dividing these microtubules. These are the two nuclei. The chromosomes have used these microtubules as a little pathway to walk away from each other, to separate the two identical sister chromatids, and eventually that's going to pinch off. You're going to have a new cell. So here we have a couple cells, a big cell and a small cell, and this is a mitochondrion in the middle here. And it needs oxygen, glucose, and it needs to release heat and waste. So say oxygen needs to come from the environment and boom, come through the membrane and travel all the way through and then eventually make it to the mitochondria. Conversely, the waste that, glucose too. Conversely, the waste that's being produced, like CO2, nitrogenous waste, so first ammonia and then other things, they need to get out. So what I want you to do is take a ruler and just measure this. Compared to this one, this little cell over here, check it out, the oxygen, boop, already there. Glucose, boop. So here we have a much shorter travel distance. Sorry about the sloppy handwriting. So shorter travel distance. This means the cell can get its supplies faster and get rid of things faster, the waste products. So that's a real critical thing you need to keep in mind. It's all about surface area to volume ratio. Here, surface area to volume ratio is high. Lots of volume, a lot of surface area for the amount of volume. Here, the surface area to volume ratio is low. Not good. This is going to back up in the system, the carbon dioxide and the other waste products, the nitrogenous waste. It's not going to get its oxygen and glucose fast enough. And this one here, sad cell. Over here, happy cell. Sad, happy. Sad, happy. Okay? Low surface area to volume, high surface area to volume. It's efficient. It can move. It's like a computer that goes boom, boom, boom. This is like a slow computer they're about ready to throw away because it's just taking forever, or an old phone, or whatever it is you've had electronically. So the larger the surface area to volume ratio, the faster the rate of the substance will move into and out of the cell. And we see some adaptations for better absorption of materials. Increased surface area, basically. So, root hair cells. Look at this. Woo! Instead of just going across like this, it has this long outward pocket. Look at all that extra surface area. That is extra surface area. That cell's going to be able to absorb water and minerals easier. Boom. Which the plant's going to need. Here, epithelial cell, small intestine. Look at this. Ooh, look at all these little fingers, just like the root hair. So think if that membrane just went like this. You're going to lose out on all this extra surface area. Guess what? This is extra places where food can get into your blood. It has to get through this membrane. It's all a function of how much membrane it's exposed to. If exposed to a lot of membrane, it'll be able to get in there. Okay? And then we have a red blood cell. Remember, it's carrying oxygen. And it needs to offload that oxygen when it gets to the cell. So you have a flattened disc, lots of surface area to volume. And again, surface area is going to help that oxygen get onto and into the cell when it goes through the lungs. Oh, so explain this. More surface area to absorb water and minerals. And interestingly too, roots, we'll talk about hyphae later, but these are fungi. it further increased surface area by associating with these hyphae that have even more surface area than these regular cells that we're looking at. And without that, a lot of trees wouldn't be able to absorb enough water in order to survive. And of course, small intestine, again, increased surface area. Well, they increase surface area volume ratio to absorb, I'll use a fancy word for food, nutrients, okay? So they can go into the blood. So they need to be exposed to the membranes of your cells so they can get in there. And then we got your red blood cells. Again, high surface area to volume ratio allows release EASE of oxygen at your tissues. When it gets to the tissue it needs to go to, it can get out. So here we have very thin, small, this is a Ascaris worm, I believe. It has a high surface area to volume ratio. Compare this shape to a sphere. If you have a similar volume, you might expect, anticipate you're gonna have a lot more surface area if you pull it out and kind of squish it down like in the worm. And any kind of circular structure is not usually going to be very big because you're going to run into that problem. Whatever's in the middle of that sphere is going to be pretty far away from the edge, an available membrane to get food, nutrients, oxygen. So if when you see large cells, say in animals, normally they're long and thin, like muscle fibers. Giraffe, I think. Giraffe has a nerve cell that's like 6 feet long, but they're long and narrow, very thin, like a hair, not like a sphere. Okay, so a really cool electron microscopic image of a red blood cell. Really cool how these kind of dip in like a donut. Again, that little dip increases the surface area so oxygen can be released. And rod cells, and this is your retina, this is the base of your retina, and you can see lots of surface area to absorb the light. Those are the rods, the white ones, and down here, look at, they're kind of shaped like this. What does that remind you of? Maybe a cone. So those are cone cells. Rod cells, they just sense any kind of light whatsoever. Tiniest, 1 photon, supposedly, they've been measured being able to pick up. The cone cells are for color vision. So color vision from those cone cells. Pretty cool. Here's how genes get expressed. You start out with DNA, and then you go through something called transcription. And that makes RNA, and that leads to translation. And just to kind of talk about this, this happens in the nucleus. This happens in your cytoplasm. And then it folds up your polypeptide and eventually becomes your protein. EIN. Okay, so we got a couple examples. They don't follow the normal idea of cell theory. So let's get into it. So recognize this as your muscle striated. These are called striations. So this would be like in your arm, your mouth, any kind of muscle that's manipulating a bone, extending it or flexing it. So the issue, the thing that makes these muscle cells interesting because they're number one, much larger than normal. Maybe if I'm writing on this red, I'll switch this. Up to 30 millimeters. That's about how big of a screen you're looking at. That's about an inch. So that's a long cell. It's a really long cell. Remember, most cells are microscopic. So that's pretty notable. And one cell can have hundreds of nuclei. Normally we think of cells having one nucleus. but this is an example of a cell that has more than one nucleus. So it's kind of weird. And remember, whenever humans, we try to put rules in things, structure, there's usually some exceptions. This is one of them. Here we have giant algae. Let me switch my color back to red. I think that'd be best. So giant algae. It's like Christmas theme with my colors here. So giant algae. And this is one huge cell can be up to 100 millimeters. So yeah, 100 millimeters. That's 10 centimeters. That definitely probably spans maybe your screen. So that's like 10 centimeters is half a ruler. Okay, so or no, like a third of a ruler. So that's pretty long. Okay, that's pretty big. And one nucleus. So this whole thing is one nucleus. And the last one I look at is aseptate fungi. And most fungi are made of hyphae. These are hyphae that I mentioned earlier. These are kind of like, they look like little roots, little branches. And usually they have, most fungi, they have septum or septate. So little, basically, divisions between sections. Well, guess what? Aseptate, that means no septates. This is all one long big cell. Okay, one interrupted tube. And that's what makes it unique.